So our first talk in Living on the Edge is from Dr. James Clugston. He's from Western Sydney University and he's a fellow of the Montgomery Botanical Centre. Is it a fellow or? Yeah. So James is a conservation biologist with broad research interest in molecular diversity and the evolution of, I can never say this one, how do you say it? Gymnosperms, yeah, and angiosperms, with a focus towards species conservation of cycads. James is passionate about cycads due to their interesting biology and unique adaptations that have allowed them to survive for millions of years. Now, the focus of his recent research is the development of new molecular techniques to better understand cycad genetic diversity and to apply this to conservation worldwide. In his work, James uses a broad range of molecular techniques and approaches in conservation genomics, systematics, phylogenetics, and plant evolution. James is also co-chair of the Global Conservation Consortium for Cycads, Australia, and he's an active member of the IUCN Cycad Specialist Group. James has recently returned from 4 very successful field trips across New South Wales, Queensland, Thailand and South Africa, sampling cycads for genetic analysis. And he is going to share conservation stories from those places. Can you all welcome James?
Thank you very much. I'm going to see how this is working. Does the later pointer work here? It does, not very well. Okay, good morning everyone. So as you probably realised, I quite like cycads. And so if anybody knows me, I've always seemed to really have an affiliation for these plants and work with them. But of course, a lot of what I'm going to talk about now is, although it's focused on cycads, it's a lot that can be applied to other groups of plants as well, because what we're really trying to do here with cycads is make a bit of a difference and make some techniques and use techniques that can be applied and with different thinking, because there's a really good focus globally on cycad conservation. And this is not something I really need to point out to you, you've got everybody here, is that worldwide plants are under threat, and this is a significant problem, especially for Australia, but not for Australia, generally worldwide really. Cycads, especially from global threats from overcollection, extinction from the wild, climate change. And although the picture just here, this is a population of Cycas meconicae, and this is through a controlled burn, but it still indicates that the damage that burns can do, especially when they're not the right time of year, or when you have invasives in a habitat that means the fires burn at higher frequency and can damage the cores of these plants, and that can be a big problem. So if you think about cycad diversity worldwide, so cycads, with the latest information, are around 330 million years old. So this is the split between the 2 major families of cycads, which is Zamiaceae and Cycadaceae. And that is a long time. And that's a lot of evolutionary history that's happened in these plants. And Australia is a real species richness hotspot for cycads. And we have 4 genera found in Australia. And we have Bauhinia, we have Lepidozamia, Macrozamia, and Cycas. So we have both families represented. And generally speaking, there's no other continent or country in the world that have both families represented of cycads in their diversity. So that really shows how Australia is important for these plants. But cycads worldwide are incredibly threatened. Actually, cycads, as far as plants go, are the most threatened group of plants by the numbers. So you have more than 62% that are threatened with either being extinct in the wild, critically endangered, or vulnerable. And this is really important, looking at the IUCN Red List. And this indicates this is a group that's really important to conserve. And this is partly due to habitat contraction, loss of environment, extinction of pollinators, seed distributors, and really poaching and overcollection is a huge problem in cycads. The poaching and overcollection, I will add, is less of a problem that we know about in Australia, but we are seeing significant problems in Australia in cycad populations of species that are of commercial value and where the seeds are being taken in masses from the wild, and none of them are staying in Australia. They're all being exported outside in Thailand, in Indonesia, or in the Middle East, because these plants hold a lot of value. So for a couple of cycads, this is a species I focus on for my PhD. This is Cycas calcicola. This is from the Northern Territory, and this species is growing in solid limestone. It's a really incredible plant. Not an easy plant to grow in cultivation, but it's a really beautiful one. So this is a really good example of Cycad or Cycadaceae. Then, of course, we have Bauhinia. This is Bauhinia species Tinaroo, which is a subjective plant. It's probably just a Bauhinia spectabilis from northern Queensland that has a serrated margin that makes it look like the other species, Serralata. But this is me and my colleague, Michael Coloni, from the Montgomery Botanical Centre. And, of course, one of New South Wales' endangered species, Macrozamia johnsoni, really beautiful, really elegant species, but from a really small distribution. And this is what really has contributed to its current status. And these populations don't really reproduce much in the wild at present. And then Lepidozamia prosciana, we all know this. Probably many of us have got this growing in our gardens at home. And it's a really nice species. This is growing in habitat. And you can see how this species is adapted to growing in really dense forests, but it's really nice. It's a beautiful one. It's an easy one to grow. And it's actually one of these species that are quite valued worldwide. And so even though this species is quite common and has a wide distribution, there's a huge export of the seeds from Australia that are taken from the wild, often legally or illegally, because for cycads in Australia, all the seeds are only governed under CITES 2. So anything under CITES 2, cycad seeds, there is no requirements for CITES import and export permits on trade. They're completely exempt from trade. Okay, so really, let's think about when we think about cycad conservation, what are some of the tools we need to really aid this? One of the things is repeatable genetic markers that can be used from herbarium specimens to fresh collected material. And this can aid us in an approach that we can use for doing this. So this is part of my current research at the moment. And this is to aim to understand diversity of these plants, but also to find an approach for population, what I call genotyping, which is identification. So if you have a plant that's found in a legal trade, you can identify it to an individual species or to a specific population. And you can create, for example, you can create from a large marker set, you can create a small marker set that's specific to particular genera, which means if this has been identified in airports, especially some South African encephalatus, not necessarily Australian species, that are completely extinct in the wild or going extinct, you can always use them to be put back into the wild if you know which population they're from in the first place. And this is, but really, we need tools for taxonomy as well. Really, we need better tools for species delimitation in cycads, because cycads are incredibly morphologically plastic, and anybody that believes they understand the morphology of cycads perfectly, I just don't believe them. And this is me coming from a lot of experience with cycads. I understand less about cycad systematics and genetics now than I ever have done. Because the more I learn, the more I'm like, what the bloody hell is happening here? You get this data and you look at the data and I'm like, that makes no sense. But it kind of does when you think about it a little bit more in detail. And really we need databases that we can use to blast DNA sequences, or even for identification databases to understand resources of cycads and understand what are the differences between species there. And really, population assessment and surveys, this is something that's really important. And I'll be honest with you, with cycads, Australia is behind the rest of the world. We are the worst country that's the most developed to have large cycad diversity, but we have the worst assessments. I'm not saying that Queensland, because Alicia's done a really great job recently at reassessing a lot of the Queensland cycads, and I'm sure they'll get pushed through soon, but we're much slower in New South Wales and Northern Territory. But this is something that's really important, definitely an area that we can move on for the future. But of course, if you want to conserve something, you have to mitigate the threats that are really imposing these populations. So here's a really good example for New South Wales. This is I want to say this is the Macrozamia, what we call the Pluonovia complex. I will say this group was actually split up into different species. But I'll tell you, if somebody shows me images of these plants, the only way I can tell you what species it is if it's from a specific geographic location. The morphology on these plants is just too variable and it's too plastic. And many species in the complex share very similar characteristics. Really, the species concepts here are just too narrow, and they're just forming very small characters. They're not really taking into account any morphological variability. And if you can understand anything that survived 330 million years, although Macrozamia is only around 80 million years old, which is still pretty old, and that's still multiple extinction events this group of plants have gone through, they've learned to adapt. And anything that's not variable in its morphology cannot adapt to different environmental thresholds. And this is very important to think about. So species really are quickly becoming threatened. And for this group of plants, in the 70s and the 80s, these plants were mass removed from land in New South Wales and Australia because they were toxic to cattle. Cattle would eat them, and they would cause a neuromotor disorder problem, which is a degenerative syndrome between the brainstem. And this would mean that basically the cows could lose complete control of the legs. And this is from, cycads have two toxins that are endemic to cycads, cycosine and macrozamin. They don't affect us directly, but they will affect what's in the gut of the animal. And so whatever the animal's eating, grasses and things, that's a big problem. However, if we eat cycad seeds, we will get affected by them as well. But it's BMAA, another toxin, that will affect us, and it's due to the breakdown of the toxin. And organisms that do eat these, like their pollinators, have been able to either recombine that toxin or just not break down the BMA, so it doesn't have a direct effect on them. But for these plants, overcollection is not a big problem because they're not the most super attractive things. I like them, but most people, they're not one you're going to go out your way for to have in a collection because it's not something that's going to be a complete standout plant. And this is a really good example of a population. And sometimes when you're finding these plants, you can spend all day finding them, and then you find one, and then you find, oh, okay, I've just walked past 100 of them. You know, this is the problem with these things. And this kind of brings us to a really new initiative. This is the Global Conservation Consortium for Cycads. And I was part of the initial ideas for this. I'm not on the actual board of the consortium, but I'll tell you a little bit about the consortium, because this is really relevant for Australia. So this was a joint initiative from the IUCN SSC Species Survival Commission Cycad Specialist Group and Botanic Gardens Conservation International. And these two bodies conformed together to create the Global Conservation Consortium for Cycads. And really, the GCC is there to kind of help cycad diversity, bring experts together, but not just scientists, horticulturalists, growers, collectors, bodies, government bodies, to come as one and then create networks of species stewards that can kind of change how we think about conservation and change and propel conservation into a new direction, especially through things like citizen science, which is something that can be really done well in Australia for these plants. So over the worldwide, there are multiple networks. So the most current network that's just started is the GCC for Australia. And then we also have the GCC for Asia just launching. So I'm traveling to China next month to present what we're doing within Australia to the Chinese, to the government, and for them to start thinking about how they can create a GCC program for China and what can be done from the rest of the world. So this is something that's really important for us as a worldwide initiative, because of course, if you want to focus on cycads, you can't just focus on Australian taxa, you've got to think about other taxa as well. And if you've got a worldwide initiative that's set up, it means that something that's been just done regionally or globally, it makes it a little bit more significant. So for Australia, it's chaired by my good friend and colleague Vanessa Hanley, is the global chair for the consortia. And then it's myself and Tim Ubergang from the World Botanic Garden Victoria. And originally it was going to just be me, but just the workload of the consortia was a concern. And I thought that I'm really a science body and we really want to have a horticultural body in this as well. Because for cycads, the horticulture is as important as it is from the science side of it. And this was really important for us. And we don't have yet a regional steering committee actually put together, but we do have a really good background of what we want to do in the consortia. And this is really what we've got together. It's kind of the aims of the thing. So we've got two main branches, research and conservation. And one of the main aims here, and this is globally in some cases as well as just for Australia, but this is development of new tools that we can use for genetics. So this is really to take genetics into the next direction for these plants. And I can go into more detail about this later, but it's not necessarily the right audience to say what we're actually doing on the genetic side of things, because it's a little bit complex, but anybody who wants to ask me about that more than can. And we also want to genotype actually all conservation significant species globally. And we already have all the samples to do that. Probably about 6 species we can't collect. Some species, for example, occurs in the random mountain in Colombia, and it's in a cloud forest, and just no botanic gardens have been able to grow this plant yet. And so we have no DNA material for this plant in particular. But Australia we'll also genotype all of the Australian threatened species, up to 3 populations of each species, and all of the New South Wales cycads as well. And we will aim to eventually go by the Queensland group as well. So this is when we say species that are important, it's not just conservation significant. We'll also genotype phylogenetically informative taxa for Australia as well, so that have specific phylogenetic positions that we can use for markers more than anything. But also support the threat assessments of cycads, so for conservation plans, build support for exchange of material, seeds and pollen and plants between different botanic gardens in Australia. And really, we want to ensure we have an ex situ capture for all conservation species in Australia by 2030. So we want to make sure we have assurance colonies for all these plants. And I'm not saying holding collections, I'm saying assurance colonies here. So this is collections that have, you come to the Meta Collections workshop tomorrow, we can go over this in more, but really what it means is having plants of known sex, variations in sex diversity, and have genetic diversity that represents what is found in the wild, so they can be used for breeding programs and species reintroduction where needed. And that's what's important. So, was that time down? 5 minutes, okay. I'm going to go a bit quicker. So, we had the first GCC meeting in Australia in August, and this was incredibly successful. We brought people together from various organizations across the world. But when we think about cycad species, I said they're very morphologically variable species, and the genetic data has shown we probably have far fewer species than we thought we did otherwise. And really, the environment is probably likely what is reflecting these morphological adaptations and not necessarily speciation. So here's an example in Cephalatus. This is one species, and this is one population. This shows the morphologically variability in this species, which is pretty incredible in a population. And this is what kind of makes you think about, you see this and you're like, okay, you have a good idea what you're going forward to looking at these species and how you kind of think about them. But you see this and you're like, okay, this doesn't make sense. It's a lot more variable than you want to put your mind to it. So really, how would you, with cycads, this is an example in South Africa, this is not Australia, and the reason why this focus here is on South Africa is because just impact. The impact of the funding and the impact of the research at the moment, and that's the truth in this. But this is a way that we're testing this varies by how would you determine species boundaries in cycads, and this is complete population sampling from individual species groups across cycads. So this is looking at 4 species from a species group, sampling the pollinators, the volatiles, the chemicals that are produced from the cones, sampling all of the genetics and looking at differentiation. This will allow us to determine what is a species in cycads and how have they evolved to different environmental thresholds, and are they differentiated by pollinators and genetically. And these are the plants here. So we have Trispinosus, Arenaris, Horridus, and Lemani. Pretty attractive plants. This is Horridus in habitat. You don't want to fall in this plant, but it's a nice plant. It is indeed. And this is Lemani. It's South Africa, but they're still incredible plants. So really, but there are a few barriers to research in Australia. Really, generally speaking, we have limited knowledge of the distribution of species. Although we have what we have in the BEM specimens, for example, somewhere in Woolamai National Park, we certainly can't say we've escaped through all of Woolamai National Park to try and find cycads. And that's the truth. We only have what we have at the moment. And there's probably a lot more populations out there. And so many of these populations are very isolated. And the access to some of these plants are often on private land. And landowners are not always the most willing to let you on the land. If you find a landowner and say, oh, I'm from a university, I want to collect DNA from your plants, you're often like, no. Oh, why? I'm like, oh, no. I'm like, okay, well, that's very helpful. And here's a case study here. This is Cycas desolata. This was a species that was only known to occur from a single population in the wild on private land. I wrote to the landowner. I emailed the landowner, phoned the landowner. We even offered to offer a fee for access and pay them for the time additionally, and completely unwilling to let us on that land to collect DNA samples and collect seed. Luckily, we did manage to find an additional population of this plant that was incorrectly identified, and there probably is another population out there as well. But we couldn't go there because I could not find the details of the landowner. I spoke to the local police station. They did not know who the landowner was and their contact details, so we just could not try and get access, and so that was it. But we did find seeds for this one plant, and these are now in development of a new assurance colony for these, almost like. And then the last one was the Cycad cupida. This was a species that is only found on one part of land and in one area, and this species is significant, but we was able to get access to this plant, and it really is a beautiful species. So to kind of conclude really, how can botanic gardens and herbarium collections support research for Cycad? So data collection is something that's really important, but also access to material. But of course, to get access to material, you need to have genetic collections. And Montgomery Botanical Center, one of my other organizations I work for as well, has the world's largest legal Cycad collection. And I'm sad to say that Montgomery Botanical Center has a significantly larger Australian Cycad collection than any botanic garden does in Australia. And they hold up to 98% of the genetic diversity for some species there. And they have collections that hold most of the gene diversity for species that can be used directly for reintroduction programs, and they are doing that. And this is something that Australia could do a lot better. Any questions?
Thanks, James. That South African example provides a bit of a entree of what we need to do, doesn't it?
Yeah, exactly.
Yeah, so on both the surveying and sampling and the ex situ conservation. So, any questions? Alan?
James, thank you. Do you have any idea of who and what the pollinators are and which ones might have been lost? Yes
and no. So for Australia, we have you have a section of thrips called Cycadofrips. Cycadofrips chadwickii is one particular species, and then you have a series of beetles as well in the family Cuculinidae. And those Cuculinidae beetles are some of the main pollinators for cycads for Australia. Species, pollinators that are going extinct for Australia, we don't really know. We don't really know there's not many populations that are not actively breeding, not actively breeding at the moment, so we haven't seen that. But of course, for South Africa, there are species that we know that have pollinators extinct. But again, the population haven't been surveyed enough, and the pollinators haven't been recorded properly yet for Australia. And the pollinators are certainly not my area. But there are some entomologists, but very few in Australia that have any focus on the pollinators.